Okay, so what I would like to do is go over this review, and I looked at a few reviews over Zinc, but I like this particular one because it phrases it in a way that I like. It starts off by saying the essential toxin. Um, Pretty much every element can be toxic, right, in the wrong concentrations. So I like this one because it's not just saying uh, take as much zinc as you can because it's healthy. It's framing it in a way that I appreciate, where it's elements, salts, these metals. You need to have respect for them because they do play a large role in your life, and as a result, you need to be mindful of how you of your relationship with them. And that's pretty much every substance. You need to respect and be mindful of your relationship with pretty much every substance that you put in your body. Okay, so I I decided to pick this one for one because it's relatively new, 2010, so it's only 10 years old, and it's free. That's another reason why I chose it. But then also their framing of the topic I do appreciate. So I don't know if I'll do this all in one go or if I'm going to uh, split it up into two parts, but let's just get into it and see where we end up. Anyways, compared to several other metal ions with similar chemical properties, zinc is relatively harmless. Only exposure to high doses has toxic effects, making acute zinc intoxication a rare event. In addition to acute intoxication, long-term, high-dose zinc supplementation interferes with the uptake of copper. Okay? Hence, many of its toxic effects are in fact due to copper deficiency. So this is something to be considerate of, is that if you are, if you have a problem, if you're showing some sort of symptoms due to your copper intake, it's probably not the result of the intake of copper, but due to a deficiency of a different ion. I'll talk more about that later. While systemic homeostasis and efficient regulatory mechanisms on the cellular level generally prevent the uptake of cytotoxic doses of exogenous zinc, endogenous zinc plays a significant role in cytotoxic events in single cells. So, the zinc that you consume, you have a lot of regulatory mechanisms to prevent yourself from uh, getting hurt from it. But on a local level, usually in a very controlled manner, endogenous zinc, or zinc within the body already, can play a significant role in the cytotoxic effects, or the cell-killing effects. Here, zinc influences apoptosis, which is programmed cell death, by acting on several molecular regulators of programmed cell death, including caspases, which are a type of self-regulating or positive feedback loop enzyme, protease explicitly, and proteins from the BLC and BAX families. One organ where zinc is prominently involved in cell death is the brain. And cytotoxicity in consequence of ischemia and trauma involves the accumulation of free zinc. So let's look up ischemia because I don't know what that is. Oh, okay. Restriction in blood supply to tissues, causing a shortage of oxygen. Interesting. Usually caused by problems with blood vessels. Okay, good to know. So the restriction of blood or trauma involves the accumulation of free zinc in the brain. Fascinating. Rather than being a toxic metal ion, zinc is an essential trace element. Whereas intoxication by excessive exposure is rare, zinc deficiency is widespread and has a detrimental impact on growth. So, yeah, that's a problem. Neuronal development and immunity, and in severe cases, its consequences are lethal. Anyways, zinc deficiency caused by malnutrition in foods with low bioavailability, aging, certain diseases, or deregulated homeostasis is a far more common risk to human health than intoxication. So here they're saying the risk of intoxication is less than the risk of deficiency. Okay, that is and that would be a part of a healthy understanding of your relationship with this particular metal. So let's begin. In the periodic table of elements, zinc can be found in group 2b, together with the two toxic metals cadmium and mercury. So these are in the same column. Okay. And nevertheless, zinc is considered to be relatively non-toxic to humans. This is reflected by a comparison of the LD50. The LD50 is the concentration or the dosage that will kill half of the people or half of the creatures that receive that dosage. So I believe it's lethal dose 50% of the sulfate salts in rats. And according to ToxNet database of U.S. National Library of Medicine, the oral lethal dose 50 for zinc is close to 3 grams per kilogram of body weight. That is more than 10 times higher than cadmium and 50 times higher than mercury. So when it comes to the toxicity and lethality of zinc concentration on a per weight basis, that is it is far less deadly, far less lethal than cadmium and mercury. Anyways, and on top of that, I believe you have more mechanisms to handle zinc ions, so you are probably going to be there'll probably be less long-term side effects from acute high exposure of zinc. Regardless, an important factor seems to be zinc homeostasis, allowing for the efficient handling of an excess of orally ingested zinc. Because after intraperitoneal injection into mice, and I believe this means, let's look this up because I don't, injection of the substance into the peritoneum, the body cavity, okay. So this is, yeah, just like where all of your organs, your what are considered your internal organs are located at. Anyways, because after intraperitoneal injection into mice, the LD50 for zinc was only approximately fourfold higher than for cadmium and mercury. So it is more toxic when it's directly injected into you. Whereas if you consume it, your body has mechanisms to help prevent the absorption of that excess zinc. Okay. In contrast to the two other metals for which no role in human physiology is known, zinc is an essential trace element not only for humans but for all organisms. Okay. So this is not just something that just mammals or just animals needs. All organisms. There's probably a few exceptions, but we might as well just say all organisms. I would say, yeah, probably all, because it's necessary, at least in most DNA replication machinery, if I remember correctly. Regardless, we'll keep on going. It is a component of more than 300 enzymes and an even greater number of other proteins, which emphasize its indispensable role for human health. And I believe this is just 300 enzymes in the human body. Optimal nucleic acid and protein metabolism, as well as cell growth, division, and function, require sufficient availability of zinc. Okay. In this review, we will give a brief summary of zinc homeostasis, and homeostasis is, what's it called? The ability to remain constant, the ability to stay at a steady state. Okay, so zinc homeostasis, followed by a description of the events of acute zinc intoxication and the consequence of long-term exposure to elevated amounts of zinc. Besides systemic intoxication, there exists evidence for physiological involvement of endogenous zinc in toxicity on the cellular level, for example, regulating apoptosis in many different cell types and having a prominent role in neuronal death. In the end, we will also briefly discuss the detrimental effects of zinc deficiency because unless they are exposed to zinc in the workplace or by accident, healthy individuals are at a far greater risk of suffering from the adverse effects associated with zinc deficiency than those from associated with intoxication. Let me see. Yeah, so they're saying the normal person is far more likely to have adverse effects from deficiency rather than getting too much. Okay. So 2, zinc homeostasis. The human body contains 2 to 3 grams of zinc. That's not a lot. That's not a lot of zinc in the body, comparatively to the total mass of your body, right? But those 2 to 3 grams of zinc play a vital role. And nearly 90% is found in muscles and bone. Other organs containing estimable concentrations of zinc include prostate, liver, and gastrointestinal tract, the gastrointestinal tract, kidney, skin, lung, brain, heart, and pancreas. Oral uptake of zinc leads to absorption through the small intestine, and distribution subsequently occurs through the serum, where it predominantly exists bound to several proteins such as albumin, alpha microglobulin, and transferrin. That's interesting because transferrin, I believe, also transfers iron. Yeah, so transferrin is an iron binding protein, but iron ions and zinc ions are similar. They're both divalent. Usually, iron can sometimes be trivalent, and I think in very rare instances, tetravalent. Don't quote me on that. But yeah, we should continue. On the cellular level, 30 to 40% of zinc is localized in the nucleus, 50% in the cytosol, and the remaining part is associated with membranes. Interesting. Cellular zinc underlies an efficient homeostatic control that avoids accumulation of zinc in excess. The cellular homeostasis of zinc is mediated by 2 protein families, the zinc importer, also known as ZIP, or ZRT, or IRT-like proteins, and this group contains 14 proteins, that transport zinc into the cytosol, and the zinc transporter, or the ZNT family, comprised of 10 proteins transporting zinc out of the cytosol. So here we have our ZIPs, which can take the zinc outside of the cell and funnel it into the cell. And then we have our zinc transporters, or the ZNTs, that take zinc from inside of the cytosol to the outside. And so the zinc cannot cross the barrier normally, right? So the barrier, the cellular membrane, is usually resilient to allowing ionic material from just passing through. Okay, that's because the cellular membrane, for one, it has an electric potential across it. So if ions could go through, they would go through very readily, and that would remove the electric potential across the cellular membrane and therefore result in cell death. Okay, so the control of ions in the cell is very important. Continuing, the same transporter families also regulate the intracellular distribution of zinc into the endoplasmic reticulum, mitochondria, and the Golgi. In addition, many mammalian cell types also contain membrane-bound vesicular structures, so-called zincosomes. I did not know that. These vesicles sequester high amounts of zinc and release it upon stimulation with growth factors. Interesting. So there are places in the body, or in the cell, in some cells, that will have organelles, or small little bubbles, where zinc will be concentrated into. I find that fascinating. And then in the rare case where the cell gets a signal to release this zinc, it does. So for instance, growth factors. So when a growth factor stimulates a cell, and growth factors are hormones. So if the cell gets stimulation in order to grow and divide, that could cause these bubbles of zinc ions to be popped or released. Fascinating, because that could mean that the zinc would be playing an important role in the let's say if the cell is producing a lot of new proteins, and those proteins are going to need cofactors and metals, metal ions to stabilize them, then the added production of zinc might be important. So this could be a way that zinc availability or zinc is stored for very important events in the cellular life cycle. Interesting. Finally, metallothioneins or MTs, they play a significant role in zinc homeostasis by complexing up to 20% of intracellular zinc. So MTs, or metallothioneins, are ubiquitous proteins characterized by a low molecular weight, so 6 to 7 kilodaltons, which is just a measurement of mass, high cysteine content, which cysteine is an amino acid, and it's an amino acid that has a thiol or a sulfhydryl group on it. And these thiols or sulfhydryl groups, the sulfur is able to react with a lot of ions and a lot of metal ions. So not just zinc, but they can react with quite a bit. So for instance, you're probably aware of fool's gold or iron sulfide. That is a metal and sulfur compound that is very stable, relatively stable, right? Because sulfur is pretty able to have strong bonds with metals. So these metallothioneins have a lot of these sulfur functional groups on them, which are useful for complexing with the zinc ions or other metal ions. So, actually, let's look this up just so that y'all can see. So here we can see this is a peptide. So it's this whole thing is a protein, and proteins are polymers of amino acids, and here you can see the three-dimensional structure of the amino acid. And these yellows are the residue, the sulfur residue of the cysteines. So remember, this is a cysteine-rich molecule or a cysteine-rich protein. And then these cysteines, these sulfur groups, interact with a lot of metals and sequester them, and then they can be used for a special function. So here we have this one of wheat that is binding. This is the wheat metallothionein, and it's binding zinc ions in this particular structure. And here we have a yeast metallothionein, and this one is bound to copper ions. And then here we have cyanobacteria, which is the blue-green algae, if I remember correctly, and this one is bound to zinc ions. Okay. So they play an important role in sequestering these ions, because remember, these ions are important. Your body needs to control the concentrations of zinc ions to maintain homeostasis. And one way you can do that is by production of these proteins that can help sequester them, they can help transport them, they can help release them given certain circumstances. anyways, continuing. Metallothioneins are ubiquitous. Ubiquitous means found all over nature. Okay, so ubiquitous proteins characterized by a low molecular, I read that, through different affinities of the metal ion binding sites. It can act as a cellular zinc buffer. So buffers are things that can help prevent wild swings in activity. So if there is a pH buffer, those pH buffers will prevent the pH from changing too quickly in your solution. Okay, so over several orders of magnitude. So large orders of magnitude of different cellular or concentrations of zinc can be modulated, can be buffered to a much smaller range using these metallothioneins. Anyways, dynamic regulation of cellular zinc by metallothionein results from the synthesis of the apiform thionine in response to elevated intracellular zinc levels by triggering the metal response element binding transcription factor. Okay, so transcription factors are proteins that bind to the DNA when they're activated. They get activated to do something. And when they're activated, they can interact with the DNA, and that can influence how active the transcription of that DNA is, or the gene that is just downstream of that transcription factor. Okay, so the transcription factors will bind to promoters or just different elements of the DNA, and that influences the gene expression of that area. So these metal response element binding transcription factors, these are transcription factors that seem to be activated by the presence of certain concentrations of metal ions. So I'm going to look this up just to make sure. I'm having a hard time, Google knows what I want. And we will do wiki because, yeah. So this is at the metal regulatory transcription factor 1, and this is just one of them. There's probably a multitude of them. And this induces the expression of metallothioneins, which is that protein that I was talking about earlier, and other genes involved in metal homeostasis, so trying to maintain the homeostasis of the body. These are all, I should say, they are influenced by this metal regulatory transcription factor 1, and there's probably more. So, and this is in response to higher concentrations of these heavy metals. Okay? The protein is a nucleocytoplasmic shuttling protein that accumulates in the nucleus upon heavy metal exposure and binds to promoters containing metal responsive elements, or MREs. So promoters are areas of DNA that are before the gene that things can bind to influence transcription rates. So, and different promoters can have different properties, and the promoters that contain the metal responsive element, which would be a series of, just like a code on the DNA, this code will bind to this metal regulatory transcription factor. So, going back to this, oh, and they luckily have a beautiful diagram, and unfortunately, I'll open it in a new tab. That should probably, okay, good. And I wish it wasn't that big. Okay, so here we see our transporters that they spoke about earlier, the ZIPs and the ZNTs, which can be one way to control the concentration of zinc in the cytosol. And then the zinc can go we spoke about this earlier, the zinc can also be put into these small vesicles as a way to control, as a way to sequester and have little packets of zinc that can be useful in certain circumstances. So that's what they call here by cellular sequestration, right? And then here we have our zinc binding proteins. So the metallothioneins is one of them, right? So zinc, when it's in the cell, it has all these cellular activities that it plays a role in, such as catalytic and co-catalytic and structural functions of the proteins. So they are binding with the proteins to function as enzyme, to function as the enzyme cofactors, or for actual structural components, because remember, these metal ions can bind to a few things around them. So by binding to a few things within a protein, it can solidify the structure of the protein. Continuing, signal transduction, that would be if there is a signal on the surface of the cell, then if that signal needs to get down into the cell, and one of the ways to get that signal down would be altering zinc concentrations. Anyways, protection from oxidative stress, and then the inhibition of caspases. So the caspases are proteins that they are proteases explicitly that can signal cell death or cause the cell to die. And this can help the BCL2 and BAX ratio, which we will talk about later. And then another thing, it doesn't just have all these cellular abilities for cellular vitality. It also has roles in cell death as well. So the BCL2 BAX ratio and the release of cytochrome C, that's a part of apoptosis. We'll talk about that later. Activation of the pro-apoptotic molecules like P38, activation of potassium channels, inhibition of energy metabolism, and induction of oxidative stress. So these would be important if, say, it was a cancer cell, and zinc would plausibly play a large role in causing that cancer cell to end up killing itself. Okay, and so here, this is what we just spoke about on the paper. Here are the metal transcription factors. Okay, so let's say you have a large influx of metal ions such as zinc, and it doesn't have to just be zinc, it could be cadmium or lead. If you have a high concentration of this zinc, what happens is the zinc will bind to the, I'll call it the metal transcription, or the MTF1s, so these are the transcription factor. It'll bind to it and activate it. And the activated one will then go into the nucleus, this is the nucleus, and bind to the promoter on the DNA. And then this will, in turn, create the mRNA, or the transcription of the DNA, and then this will result in the production of these, they have it called T, I forgot what T stands for exactly, but this is a protein, that ends up getting cleaved into metallothionein, so this protein right here. So this ends up getting turned into metallothionein, and then this can help regulate the heavy metal ion concentration in the cell. So we'll probably come back to this image. And yeah, T was, thionine. The apoprotein, so the protein before it's turned into metallothionein. Okay, so continuing, exposure to zinc. There are three major routes of entry for zinc into the human body: by inhalation, through the skin, or by ingestion. Each exposure type affects specific parts of the body and allows the uptake of different amounts of zinc. Okay, so we'll probably get back to this later. So, exposure by inhalation. Inhalation of zinc-containing smoke generally originates from industrial processes like galvanization when you put metal on the surface of another metal to help prevent corrosion, primarily affecting manufacturing workers. In addition, military smoke bombs contain zinc oxide or zinc chloride, making soldiers a group in which several cases of inhalation of zinc-containing fumes were described. For example, these people reported a case of 2 soldiers who developed adult respiratory distress syndrome, or ARDS, and I think that might have been changed into acute respiratory distress syndrome, which would also be ARDS, upon exposure to zinc chloride-containing smoke bombs. Yeah, it doesn't sound like a good idea, but continuing, the 2 men died 25 and 32 days after the accident. So a significant amount of time after the exposure of breathing the zinc fumes in caused it. But it might not have necessarily been the zinc in of itself. So we'll continue. Another soldier was exposed to concentrated zinc chloride for several minutes during military training. He also developed ARDS 48 hours after exposure, and then after intubation and mechanical ventilation for 8 days, he left the hospital, and 4 months after the incident, he returned to work without any respiratory disorder. Right, so not everything will kill everyone, right? Anyways, there are a few additional reports of related incidents with smoke bombs having similar effects on the respiratory tract. However, in none of these incidents, there was unequivocal evidence that zinc was the main cause for the respiratory symptoms. Not only was no information about the concentrations available, but also the inhaled smoke contained several other ingredients besides zinc chloride. In addition, zinc chloride is generally caustic, so the effects could have arisen from the specific properties of the compound rather than being a direct effect of zinc intoxication. So it is possible that it wasn't the zinc in of itself, but the just the chemical reactivity of salts in general, or salts like zinc chloride in general, could have also caused this issue, right? And there are plenty of other chemicals in these smoke bombs that could have also caused issues. So who knows what the actual, if that was actually the case. Anyways, the most widely known effect of inhaling zinc chloride smoke is the so-called metal fume fever, or MFF. And this is not just for zinc, this is for a lot of different metals. And this is mainly caused by inhalation of zinc oxide. This acute syndrome is an industrial disease which mostly occurs by inhalation of fresh metal fumes with particle sizes of less than 1 micrometer. In occupational situations such as zinc smelting or welding, symptoms of this reversible syndrome begin generally a few hours after acute exposure and include fever, muscle soreness, nausea, fatigue, and respiratory effects like chest pain, cough, and dyspnea. That's dizziness, right? Oh, shortness of breath. Okay, yeah. The respiratory symptoms have been shown to be accompanied by an increase in bronchial leukocytes. So these are white blood cells in the bronchial. I forgot the actual word for it. In general, metal fume fever is not life-threatening, and the respiratory effects disappear within 1 to 4 days. Good. And remember, you can get metal fume fever from a lot of different metals, not just zinc. Okay. So this isn't explicitly a zinc thing. This is, you need to have respect for all chemicals that can have some sort of physiological effect. Right? Anyways, development of metal fume fever is connected to the exposure level, but very little data is available concerning the zinc concentrations that trigger the syndrome. 2 volunteers developed metal fume fever as a consequence of acute inhalation of 60 milligrams of zinc per cubic meter as zinc oxide. So every cubic meter had 600 milligrams of zinc in it, and then as these people were breathing that air, 2 volunteers were breathing that air for 10 to 12 minutes, they got a certain amount of that into their lungs. So these people reported about workers. Oh yeah, and then, wait, oh, so those were volunteers who volunteered to breathe that air. Science is weird, man. Anyways, Hammond and colleagues reported about workers who had shortness of breath and chest pain 2 to 12 hours following exposure to this concentration as zinc oxide in the air. Only small changes in forced expiratory flow were observed after exposure to 77 milligrams per zinc per cubic meter as zinc oxide. So as you are lowering the concentration from 600 to 320 to 580 to 77, there are less and less effects of the metal fume fever. Anyways, several reports of exposure to lower concentrations of zinc oxide for up to 3 hours, and this concentration for 6 to 8 hours did not result in symptoms of metal fume fever. So even lower amounts, even for longer times, are much easier to deal with. Today, the permissible exposure limit, according to OSHA, is 5 milligrams per meter for zinc oxide dust in workplace air during an 8-hour workday and a 40-hour work week. Dermal exposure. So dermal absorption of zinc occurs, but the number of studies is limited and the mechanism is still not clearly defined. Agren and colleagues pointed out that the pH of the skin, the amount of zinc applied, and its chemical speciation, so what are the anions, what type of salt of zinc is it, all this influence the absorption of zinc. In a study in which 25% zinc oxide patch was placed on human skin for 48 hours, there was no evidence of dermal irritation. Okay. In another study, comparing the dermal effects or the skin effects of different zinc compounds in mice, rabbits, and guinea pigs, zinc chloride was clearly the strongest irritant. So, but keep in mind, zinc chloride is kind of caustic in general. It's a somewhat caustic chemical, somewhat acidic in of itself. So that might not be from the zinc itself. It could be from the relatively reactive nature of zinc of the chloride anion of zinc, and then followed by zinc acetate causing moderate, and zinc sulfate causing low irritations. Consistent with the study by Agren, zinc oxide did not show any irritant effects on the skin, and that's probably because zinc oxide is very, very insoluble as well. Anyways, as mentioned above, zinc chloride is caustic, and irritation does not necessarily indicate a toxic effect of zinc. In contrast to a potential harmful effect of zinc on the skin, it should be noted that zinc is a well-known supplement for topical treatments of wounds and several dermatological conditions. So we sometimes use zinc as a way to treat the skin. For instance, pyrothone zinc is a way to treat dandruff because the pyrothone zinc is able to kill a lot of fungi. So by applying it, you end up allowing the scalp to grow in a healthier manner. Anyways, based on the existing data, it can be concluded that dermal exposure to zinc does not constitute a noteworthy toxicological risk, which is good. So, oral exposure. Due to its nature as an essential trace element, oral uptake of small amounts of zinc is essential for survival. The recommended dietary allowance, or the RDA, for zinc is 11 milligrams per day for men and 8 milligrams per day for women. Lower zinc intake is recommended for infants and children. because of their lower average body weights. This is significantly below the LD50 value, which can be estimated to be 27 grams of zinc per day. Oh my gosh. Okay, 27 grams. I was like, wait a second, that's not that much greater. But no, so don't confuse the milligrams with grams. So this is, you know, over 2,000 times greater concentration. Whew! I was like, wait, that's not a. In general, well, shoot, 27 grams of zinc in a day. That to only kill half of people. That's a ridiculous amount. Man, in general, uptake of such an amount is unlikely because approximately 225 to 400 milligrams of zinc have been determined to be an emetic dose. So what is something that is emetic? I believe that means that you puke. Yeah, so it causes vomiting. So if you get even this much zinc in your body, you're going to puke it out, right? You're going to get so sick of it, you're just going to puke. Interesting. However, there is one published report of a woman who died after oral intake of 28 grams of zinc sulfate. Remember, this is 28 grams of zinc sulfate, and sulfate is also playing a role in the weight of that salt. So this would probably be equivalent, if I'm understanding this correctly, it's probably equivalent to about, I don't know, 11 grams of zinc sulfate, so less than the LD50. But after ingestion, she started vomiting and developed tachycardia. I think that's abnormal heart rhythms. Oh, yeah, makes your heartbeat more than 100 times per minute. Okay, so high blood pulse rate, as well as hyperglycemia. So her blood sugar went up a lot. She died 5 days later of hemorrhagic pancreatitis and renal failure. So her pancreas basically hemorrhaging in the pancreas, and her kidneys gave out on her. Anyways, immediate symptoms after uptake of toxic amounts of zinc include abdominal pain, nausea, and vomiting. Additional effects include lethargy, anemia, and dizziness. Particular effects of excessive oral zinc exposure are discussed in detail below, but just keep in mind, this woman put grams of a zinc salt into her. That is a ridiculous amount. Okay. The gastrointestinal tract is directly affected by ingested zinc before it is distributed through the body. Therefore, multiple gastrointestinal symptoms after oral uptake of zinc have been reported. These people described several cases in which high zinc ingestion resulted from storage of food or drink in galvanized containers. So remember, galvanized, we were talking about this earlier, this is when the metal is coated with zinc in order to prevent it from corroding. So if you store food in galvanized containers, some of that zinc can leach into your food. And ingestion was caused by the moderately acidic nature of the food or drink, enabling the removal of sufficient zinc from the galvanized coating. The resulting symptoms include nausea, vomiting, epigastric pain, abdominal cramps, and diarrhea. In a study by these people, symptoms such as abdominal cramps, vomiting, and nausea occurred in 26 to 48 healthy volunteers following the ingestion of zinc sulfate tablets containing 150 milligrams elemental zinc for 6 weeks. So this is a lot of zinc, okay? However, similar doses have been used in several other zinc supplementation studies without comparable side effects. So, wait, these people. So about half of the volunteers experience intestinal discomfort, specifically from zinc sulfate. So that might be due to the fact that, and 150 milligrams, that is somewhat a significant amount. So the typical zinc supplement will have 50 milligrams of zinc in it. So this is quite a bit, 3 times that. So that would be about 1,500 times the daily recommended dosage. And it's in the zinc sulfate form, and sulfates are very soluble, so it could have caused discomfort simply because of how soluble the zinc ions were. So it might have rushed into the system rather than slowly releasing. That might be the issue of this particular study. In addition to zinc sulfate, and yeah, in addition to zinc sulfate, other zinc compounds like zinc oxide and zinc gluconate also have similar impact on the gastrointestinal tract. A 39-year-old man showed nausea, vomiting, and abdominal pain 6 hours after ingesting 150 grams of a 10% zinc. 150 grams of a 10% zinc oxide lotion, but without signs of systemic toxicity. I don't mean to laugh. Who knows what the story was behind this? So 15 grams of zinc oxide. Okay, that's a lot. Okay, that's a. Oh God, I don't know how you would be able to stomach that. But there was no signs of systemic toxicity. Furthermore, he developed gastroduodenal corrosive injury. So in the duodenum, since zinc can be corrosive in high concentrations, it ended up injuring his duodenum. And the symptoms persisted for 3 days, and on the 5th day admission, the corrosive injury showed regression without cicatrization. I don't know what that means. So is the contraction of fibrous tissue formed at wound sites by fibroblasts, reducing the size of the wound while distorting tissue. So this would be the presence of scar tissue that causes improper shapes. Interesting. So to heal without that would be good. Zinc-induced copper deficiency. So taking up large doses of supplemental zinc over extended periods of time is frequently associated with copper deficiency. This correlation seems to be caused by the competitive absorption relationship of zinc and copper within enterocytes, mediated by metallothionein. So enterocytes are cells in your intestines, if I remember correctly. Yeah, so they are the epithelial cells with all these little, I don't know what to call it, finger-like projections on. So yeah, the brush border cells are different though, if I remember correctly. Or is that the whole thing is the brush border? Yeah, so they have all these little finger-like projections on them, and that helps you absorb stuff. So here's a demonstration. Anyways, there's a competitive absorption between zinc and copper within enterocytes mediated by metallothionein. So metallothionein is this is the protein that we spoke about earlier that has a lot of sulfur groups on it. It can bind, it is in high concentrations within these enterocytes, and they're used for the absorption of the metal cations from the intestine to put it into the blood, right? So if you have a lot of zinc in the intestines, the zinc could be taking up the areas in the metallothioneins, leaving little room for copper to be absorbed. Anyways, the expression of metallothionein is upregulated by high dietary zinc content, and metallothionein binds copper with a higher affinity than zinc. Consequently, available copper ions are bound by metallothionein, and the resulting complex. Oh, I had that backwards. So, and the resulting complex is subsequently excreted. Let me. Interesting. So, I have this a little backwards. So the metallothionein, it's probably doing multiple functions. Regardless, so the metallothionein is expressed in a higher concentration due to zinc, because zinc can upregulate. Remember this photo here. The zinc can cause the upregulation of metallothionein, right? So you can get more metallothionein in the system. And if you get too much of it, then it ends up binding with the copper, and then have some of that copper get excreted. Okay, that's interesting. That's one of the roles. And I believe that would also influence iron and other divalent cations, possibly transition metal divalent cations. Regardless, and metallothionein binds copper with a higher affinity than zinc. Consequently, available copper ions are bound by metallothionein, and the resulting complex is subsequently excreted. This person and this person stated that the dietary intake of different doses of copper and zinc did not significantly alter the absorption of other metal, as long as they were given at the same ratio. Oh. Okay, irrespective of 1 milligram per kilogram copper and 5 milligrams per kilogram of zinc, or up to 36 milligrams with 180 milligrams of zinc were given. So this ratio is going to have the same ratio of absorption as this ratio, as long as the ratios remain constant. Nevertheless, copper absorption is depressed when zinc is given in high excess over copper. So the more zinc you have, the more excretion of copper you see. Interesting. Furthermore, Irving and others reported the case of a 19-year-old woman who was supplemented with 2 doses of 50 milligrams of zinc per day as part of a treatment of Hallervorden-Spatz syndrome, leading to a total dietary intake of about 121 milligrams of zinc per day for more than 5 years. And this corresponds to about 15 times the recommended daily allowance. Her daily intake of copper was 2 milligrams, which is approximately twice the daily recommended allowance. And as a result, she was markedly anemic and had severe neutropenia. So what is neutropenia? Having low level of neutrophils, and these neutrophils are white blood cells that are in the blood. So copper plays an important role in their expression and growth. Anyways, zinc-induced copper deficiency was confirmed by elevated serum zinc and low copper, and ceruloplasmin serum levels. I do not know what ceruloplasmin is. So this is a peroxidase enzyme that in humans is encoded by the CP gene, and this is the major copper-carrying protein in the blood. So remember, metallothionein is only one of the many types of proteins that exist in the body that are used to bind to metals. So there's a lot of redundancy in every system you deal with, because redundancy leads to robustness, right? So, and I believe cerulo is probably like the Greek word for copper, probably for the blue color that is associated with it. In fact, copy, oh, etymology. So it's Latin, not okay. You know what? Let's look up cerulean. Okay, just meaning sky blue. But I assume that copper is in the same. Since copper is a lot of copper salts are blue, that might be how the names came together. Anyways, 4 weeks after zinc therapy was stopped, all hematological, blood-related and trace metal parameters showed strong trends towards normalization and were normal after 8 months. So all of these blood symptoms that we were seeing, the anemia, the severe neutropenia, those went back to normal after 8 months of stopping the zinc therapy. So Prasad and colleagues reported several cases of patients with sickle cell anemia who received this much zinc, 150 milligrams of zinc per day, and consequently showed low plasma copper, low ceruloplasmin, which is, remember, that protein that transfers copper around or binds to copper, and leukopenia and anemia. Another case report described a 31-year-old schizophrenic man who had been ingesting coins for 10 years. He entered the hospital with symptoms including nausea, vomiting, and abdominal pain. Furthermore, profound anemia, neutropenia, and virtually absent serum copper and ceruloplasmin levels, together with elevated zinc levels, were diagnosed. Upon X-ray examination in a large number of coins, they had to put the dollar amount, but $22.50, were identified and surgically removed. So following the surgery, anemia and copper deficiency rapidly resolved. His copper deficiency was attributed to the ingestion of pennies, which since 1982 are composed of 98% zinc and 2% copper. Several additional reports of zinc-induced copper deficiency leading to anemia and several other cytopenias were reviewed by Fisk and colleagues. I'm going to look up cytopenia just to... So yeah, cyte just means a cell. So cytopenia means just a low number of any one of the cells that are in the blood. Anyways, the mechanism by which copper deficiency induces anemia is based on the requirement of copper for several enzymes involved in iron transport and utilization, and therefore in heme synthesis. So remember, these metal ions that I said earlier, they play an important role in a lot of different proteins. And so we have different ions for different functions. And copper, one of its functions is to bind to several enzymes that are involved in transporting iron and using that iron. So, and iron is another metal ion that is needed for a lot of different enzymes and proteins in the body, like hemoglobin. Anyways, for example, ceruloplasmin is a ferroxidase that binds copper and converts ferrous into ferric ions, allowing it to bind to transferrin and be transported. So remember, the ceruloplasmin that the copper binds to, it's a ferroxidase. It'll oxidize iron ions. And so, excuse me, it'll take the ferrous or ions or the iron 2+ ions and then remove an electron to turn it into a ferric ion. And then it's the ferric ions that are able to bind to transferrin, or a protein that transfers iron throughout the blood, and this allows it to be transported around. Anyways, cytochrome C oxidase is also dependent on copper, and this is an oxidizing enzyme that is used to add oxygen and to a lot of chemicals in the body, which can help neutralize them or make them more soluble to be transported out of the body. And you know what? I'll look up cytochrome C oxidase. So it's a large huge, actually, transmembrane protein complex found all over nature. And, oh yeah, it's also part of the respiratory electron transport chain of cells in the cellular membrane. And so what it does is it helps your body produce energy from oxidizing the, by oxidizing food, sugars, all that. And it does it by taking the electrons that come from the food, and it puts it into the oxygen to turn the oxygen into water. So it takes the electrons, it takes protons in solution, and oxygen, and makes water out of it. And that is necessary to balance the chemical reactions where your body got energy from. And let me see if there's anything else that... No, I don't think so. Regardless, this enzyme is dependent on copper, okay, and it is required for the reduction of and is required for the reduction, so turning the ferric ions into ferrous ions. So it can take an electron and add it into this ferric ion to turn it into a ferrous ion. And then the ferrous ions are used in hemoglobin. And so if you have a buildup of ferric ions, the hemoglobin cannot utilize it. Okay. Anyways, in addition to interference with heme synthesis, copper deficiency causes approximately 85% reduction of ESOD in the red blood cell membrane, decreasing the red blood cell surviving time. So what is ESOD? Actually, extracellular superoxide dismutase? Okay, one second. Let me look this up. Yeah, okay, sorry. It is superoxide dismutase. I wonder why it's... Maybe, oh, that might be erythrocyte superoxide dismutase. That I'm not quite sure. But superoxide dismutase is an enzyme that will take superoxide, which is a free radical that circulates throughout the body from different types of respiratory, what would you call it? It's just a free radical that can cause damage to your proteins and your genetic material. So you have the enzyme superoxide dismutase, which requires, one of its components that it requires is copper. It takes, and it will turn the superoxides into hydrogen peroxide, which is still pretty damaging, but not nearly as reactive as the superoxide radical. And then there's another, yeah, we'll leave it at that. Anyways, copper deficiency will have you only have about 15% of the superoxide dismutase on the red blood cell membranes, and so the red blood cells will not live for nearly as long as they normally would. So this is interesting. That might play a role in something. Whereas a recent meta-analysis found no general effect of zinc supplementation on serum lipoproteins, it may occur as, and these are proteins that have fat associated with it. Actually, lipoprotein effects of zinc on plasma lipoprotein. So I think this would be like the HDL and the LDL cholesterol stuff, but let me look this up real quick Yeah, primary purpose is to transport hydrophobic lipids, also known as fat, in molecules of water. So that's what these lipoproteins are doing. So zinc has no general effect on lipoproteins. Um, it may occur as a consequence of disturbed copper homeostasis. So copper deficiency is related to alterations of serum cholesterol levels. So it might not be the zinc that's causing the issue, but zinc can influence the absorption of copper, and that can result in different types of deficiency pathologies, right? In healthy men, a daily intake of 160 milligrams of zinc per day decreased HDL cholesterol, which a lot of people refer to as the good cholesterol, probably because it's more associated with a healthier lifestyle. Also, young women who ingested 100 milligrams of zinc per day showed a reduction in HDL cholesterol. So, oh yeah, they both showed a reduction. Okay, sorry about that. A study with 24 men who were fed omnivorous diets that were deficient in copper and high in zinc uh, showed low plasma copper, low superoxide dismutase, and low HDL cholesterol, while LDL cholesterol was elevated. The study was stopped after 11 weeks because 4 participants experienced cardiac abnormalities, so that's not good as well. You do not want to have that happen during a study. So Cleve and colleagues fed one man an omnivorous diet, providing zinc to copper ratio of over 16 for 105 days, and plasma copper and ceruloplasmin decreased, whereas total cholesterol and LDL cholesterol increased. This experiment was ended when arrhythmia was detected. So there we go, cardiac abnormalities again. Taking into account several additional studies, Sandstead suggested that cardiac abnormalities were associated with zinc-copper ratios of greater than 16. Yeah, this is a long one. So, I think I will. I'll stop. I'll stop now. I'm hot. So we'll continue here at zinc supplementation and cancer later. But yeah, this whole review, pretty good. I enjoy it. I'm learning a lot. So, and remember, this is not my field exactly. This is a lot more medical than what I'm used to. So, if you notice any corrections that need to be made, leave them down into the comments, and I will see you tomorrow.